Right. I'd like to start the talk by just sort of mentioning that to date, despite well over 100 years of work in viruses, we've probably described far less than 1% of the viruses that are out there. In fact, it's probably more like 0.0001% of the viruses out there that we've described. Now in birds, we're sort of approaching our 100-year anniversary of the first viruses that have been described in birds. But if we think about how this has been done for the last 100 years, there's quite a large bias in that for the most part, what's happened is that poultry in particular have either died or become sick, cost the farmers money. And then we've wanted to know what's the cause of that, such that we go in and we look for the agent causing disease, and usually what we would find then would be a virus or a bacteria or things like that. And so in this figure here, what you're looking at is the number of viruses described sort of per year and then a cumulative number. And almost everything here in this sort of before 1995 bar are viruses of poultry that cause disease. It's really only in the last 10 years that we've turned our attention to wild birds and seemingly healthy birds to start looking at what viruses they may have. So today, when we think about viruses of birds, the things that we actually know about are viruses in poultry. Now, ironically, shorebirds are one of the more studied species or taxa for viruses because they may be infected with viruses like avian influenza. But despite that, I would still say that shorebirds are vastly undersampled as hosts for viruses, and we know very little about the viruses that infect these birds. So sort of how the talk is going to go is I'm going to highlight 3 virus species that we've done a bit of work on in shorebirds, talk a little bit about some of our more recent work in uncovering viral diversity in shorebirds, and then at the end I just want to talk about the ramifications of viral infections in shorebirds. And I thought I'd start this talk with avian coronaviruses, mostly because everybody is talking about coronaviruses these days. So what you're looking at here is a phylogenetic tree, or a tree of relationships of all the members of the coronaviruses that are out there. So coronaviruses is a family. It's very large and broad family. And if you look at the tree, you can see that there are sort of 4 groups here. So alpha, beta, gamma, and coronaviruses, those are the genera of coronaviruses. And COVID-19, SARS coronavirus 2 that everyone is talking about sits right over here. It's a beta coronavirus. Now, if you look at the little round circles there, those circles are colored by whether or not the virus is a virus of birds or of mammals. So in blue, we see that alpha coronaviruses and beta coronaviruses, these are viruses of mammals, but both gamma coronaviruses and delta coronaviruses, these are viruses of birds, okay? So essentially, there is a really, really large diversity of coronaviruses that are found in birds. Most of these viruses don't cause disease. In fact, interesting coronavirus tidbit is that the first ever described coronavirus, called infectious bronchitis virus, described in the 1920s, was from a bird, from a chicken virus. And it's really only been since 2008 that we've turned our attention to wild birds to look at what coronaviruses might be there. Now, we've done a little bit of work in shorebirds here in Australia. We had a study come out about 2 years ago where we took all the samples we would normally collect for influenza virus and screen them using a general screen to look for coronaviruses. Okay, so we didn't specify what kind of coronaviruses, just coronaviruses. So in this figure, we have our 4 species that we looked at: curly sandpiper, sharp-tailed sandpiper, redneck stint, and ruddy turnstone. And here on this axis is the prevalence. So of all the samples we collected from that species, what proportion were positive for a coronavirus of any kind? And then the bars and the 95% confidence intervals. Now, what you can quickly see is that the prevalence of coronavirus is actually quite high in shorebirds. It ranges from about 10% in redneck stint to almost 30% of ruddy turnstones. Sort of the only outlier there are the sharp-tailed sandpipers, for which we didn't find any. But that's quite an incredibly large number if you really think about it. So in 1 year of samples, almost 30% of ready turnstones were positive for some kind of coronavirus. Now, what we can do then is we can take the samples that are positive and do some genetic testing. And from that, we can understand what kind of coronaviruses they have. So here's a little pie chart. So of the 96 samples that were positive for coronavirus, a large proportion of them we didn't genetically type. This requires quite a lot of work. And in this study, we looked at more species than just coronaviruses. But a third of those were gamma coronaviruses. So that's the genera, the genus. And of that, they sort of came back as this one species here called duck coronavirus 2714. This name is quite unfortunate because this coronavirus is certainly found in more than just ducks. It's just that the first full genome came from a duck. A small proportion of them were delta coronaviruses, and we actually found 2 different species of delta coronaviruses, which I'm just going to talk about here in the next slide. And so both of these delta coronaviruses are actually novel. They're not described, they're not ratified, so they're not in the book of viruses yet. And so the first one I've called novel wild bird delta coronavirus. So sort of in this little schematic here I have on the side, we have a little pink circle showing the virus, and then all the different hosts from which we have found this virus so far. So this includes penguins in Antarctica, pigeons and falcons from the United Arab Emirates, and gulls from Europe. And we actually found it in a sharp-tailed sandpiper in Australia in a different study from the one I showed you before. So we have here a virus that's not been ratified, it's not in the books yet, but it's quite widely distributed and is found in a number of stratiforms, so shorebirds and gulls. The other virus species that we found, I've called Shorebird delta coronavirus. Again, it's not in the book of viruses, this is just the name that I've given it so far. And we gave it this name because to date, it's only ever been found in shorebirds. And so off this little tree here, you can see the pink is in 2 colors. The dark pink there, those are sequences or viruses that we found in Australia. And the light ones there, those are viruses from elsewhere in the world. And so you can see that the vast majority of sequences or viruses that's in this species that's been described have been described in shorebirds in Australia. Now, this is all very interesting, but this is where the coronavirus story stops. So because it's a virus, these viruses in wild birds don't necessarily cause disease, very little interest, and it costs money. This is all that we really know about these viruses. We know very little of the ecology. We have no idea whether our results are reflective of other parts of the world. We have no idea whether there are certain seasons that are more important than others. So maybe it's a seasonal virus in birds. We don't really know that. We know it's found in some shorebird species, so those which we've tested, but what about other shorebirds? Are they interconnected globally? Are birds moving these viruses around? Are there age effects? We really have no idea. And so this is an example sort of a virus family that we've started working on and have a little bit of data, but it really shows that we know very little about it, other than we know it's there sometimes, but we're not sure when. And so the next example I actually want to talk to you about is the virus for which we have by far the most data, and that is low pathogenic avian influenza A viruses. So just to give you a brief introduction on this virus, there are a number of ways in which we sort of name and classify this virus. So within influenza A, we subclassify it. And so the first way is based on this HA-NA system. So on the outside of the virus are some proteins that stick out. One is called the HA, one is called the NA. And so when you've read the papers about bird flu H5N1, all it means is it's type 5 of the HA and type 1 of the NA. And wild birds have a very large diversity of these. So there's 16 different HAs in birds, 9 different NAs, so that's 144 combinations that could exist in birds. Now, the second form of classification is this low pathogenic versus high pathogenic. And I'm going to start talking about low pathogenic. So low pathogenic viruses could be any of the subtypes, any of the HA-NA combinations. And these viruses are quite common in wild birds, and they don't necessarily cause disease. So we could say that these viruses are part of the natural virome of wild birds. They are sometimes found in poultry, And in poultry, they may or may not cause things like egg drop, so sort of where chickens lay fewer eggs, but for the most part, they don't really cause disease in poultry at all. So this is quite normal. Now, the reason we work on low pathogenic viruses is because of the risk of highly pathogenic, which I'm going to talk about a bit later. But essentially, we've been doing research on low pathogenic avian influenza viruses for a few decades now. And from that, we have quite a good understanding of the host reservoir. So for the most part, we know that dabbling ducks are really, really key to the maintenance of these viruses, such that if you go to a location in the Northern Hemisphere where they are sampling ducks, and you go in the autumn, on any given day, you'll find that about 20% of the ducks will be positive. Some days a bit more, some days a bit less, but almost all the ducks are positive. And if we do studies on antibodies at the end of the autumn to see how many of the ducks have had some kind of influenza in the last few months, you'll find that almost 100% of ducks will have been infected at least once. In fact, ducks may be infected and reinfected. We know that it's sometimes found in other waterfowl. Gulls are really important in maintaining sort of two of these subtypes, but where shorebirds fit in some ways is still a bit unclear. So in sites like Delaware Bay, we know that you can go in the spring and sample shorebirds, especially ready turnstones, and you'll get quite a high prevalence of this virus in these birds. Again, it's not making the birds sick, but it's there. Now, if we sample in Delaware Bay in the autumn, we don't really find a lot in shorebirds. And if we sample at other sort of key migratory sites for shorebirds, we tend not to find very high prevalences either. So in North America, for example, if you look at the Copper River Delta, which is in Alaska, so the other side, it's very difficult to detect influenza viruses in general. So whether shorebirds are important parts of the reservoir in general, or only in specific locations, is one of those things we surprisingly do not have a good handle on. Now, despite this, we've been sampling shorebirds in Australia for 9, almost 10 years. And the work has largely been led and driven by Marcel Claassen from Deakin University. So what you're looking at here is a list of the shorebird species we've sampled. And they're organized sort of in their sort of by genetic relationships. Okay, so this is just a little phylogram here to show you how these birds are related to each other genetically. And so, for example, redneck stint and curly sandpiper are really closely related, and sanderling is the next closely related virus. And so this figure shows you virus prevalence. So if we go and test the bird for viruses, which proportion of the viruses, or which proportion of the birds are positive for viruses? So that's what the bars show. They're colored by the host family. And then we also have the 95% confidence intervals. And what you can see quite quickly is a fairly large amount of red on this figure. So those are the Scolopacidae. So that is, if we go and sample the Scolopacidae, we'd probably find that they are reservoirs for avian influenza here in Australia. Particularly important species are ruddy turnstone, redneck stint. We also find sharp-tailed sandpiper and curly sandpiper. There are a few interesting sort of trends in this plot that we're still trying to get a good handle on. For example, we do find viruses in red knots, but we almost never find viruses in great knots. However, the confidence intervals on red knots are quite high. Sanderlings are another really interesting one. Even though we may catch sanderlings and ruddy turnstones on the same beach, we'll tend to find viruses in ruddy turnstones, but almost never in sanderlings. And this trend is actually seen in Delaware Bay as well. They can go sample an array of shorebird species in the spring migration and almost never find viruses in sanderlings. And we have no idea why. The other one that pops up here are avocets. Now, these viruses, or these birds, will shed these viruses for reasonably short periods, okay? So a bird may only shed virus for a few days, such that if we go and sample a week too early or a week too late, we may not be able to detect the virus. So another tool that we have in our backpack is serology, such that when you or I are infected with the virus, we're going to produce antibodies, and we're going to have those antibodies in our body for, a period of time. It may be a few months, it may be many, many, many years. So we can harness that same thing in shorebirds. So the plot is essentially the same as last time. Sort of the shorebird species here are listed the same as before. But the bars now show you the proportion of birds that have antibodies. So these are not birds that are infected now, but they're the proportion of birds that were infected in the past. So obviously the percentages here are much higher because rather than looking at a snapshot event, sort of a short-term thing, we're looking at a longer-term thing. Now, some of our observations remain. Again, there's quite a lot of red on this figure, so Scolopacidae remain really important. Ruddy turnstone, again, really, really important. Sharp-tailed sandpiper, red-necked stint, crucial. That sort of funny little relationship between the knots is still there. Don't know why. Confidence intervals on red knots remain high, but great knots quite tight. And the Sanderling story remains the same, you know, sort of like less than 1% of Sanderlings will have antibodies. So they are exposed, but they seem to not be exposed a lot to this virus. Girardiidae do pop up a little bit. The dotterel seroprevalence is high, but the confidence intervals are enormous, so it's a bit hard to say. But again, sort of the avocets and stilts pop up as well. So from this, we are getting quite a good idea of which shorebirds are important as reservoirs for this virus in Australia, but also through seroprevalence, which tells us in the past. This tells us about which viruses are important in our flyway as well, because these birds that have antibodies are not necessarily infected in Australia. They may have been infected along their migration or on their breeding grounds and still have antibodies when they come back to Australia. And so this is hugely informative and very, very helpful. Now these plots here, they bring together many years of data into a single data point. But in some of our study sites, we've been sampling the same species at the same location for many, many years. And one example is ruddy turnstones. So we've been sampling ruddy turnstones in a place called King Island now since 2013. King Island is an island off Tasmania. Really, really interesting and beautiful place. And so how this figure works is down here, you have sort of the sampling dates and year. So we usually go for 10 days, catch a lot of turnstones, sample them. Each point there, that's the prevalence of that sampling event. So that's the proportion of birds at sort of that sampling trip that were positive. We have 95% confidence intervals as well. Here in gray are occasions where we actually manage to detect virus, so we can isolate and pull the live virus out and characterize what kind of virus it is. So remember I told you before about the HA-NA system. So we find H6N8, H10N8, H3 and H12. There are long periods here for which we don't really detect any influenza. And interestingly, we also have antibody data for these birds that we can look at. And we find that in those periods here, so sort of between February 2015 and March 2017, antibody levels in the population are also really low, which suggests that these birds were not infected in a period when we were not there. So we didn't necessarily miss those infections. So this data is quite interesting because it shows that shorebirds are a bit different from ducks, whereas in ducks we have consistent annual trends where you can go back at the same time on any year and get sort of a similar results. Here in shorebirds, it looks a little bit more boom and bust. So we can go and a large proportion of birds are positive and then they may be negative again for quite a long time. This may be an island effect because we're working on a small island, because the results from Delaware Bay are a little bit different. But I'd say we still have quite a lot to learn. Now, using genetic data from the viruses, we can actually show that shorebirds are moving these viruses along our flyway. So a slightly more complex figure, but I'll just sort of work you through it. So the colors here on the figure relate to sort of the world. I've done Australia in pink and sort of the Eurasian continent and Africa there in blue. And so this is the name of the virus. It sort of tells you the species here and the location. And then the tip bit here lines up with the date, okay? So we can follow this lineage of viruses through time quite accurately. So the first time this lineage was detected, it was detected in Australia in the 80s. It stayed in Australia for quite a long time, but we didn't really sample a lot of shorebirds between the 80s and sort of in the 2000s. And here we have a sort of what we call a bifurcation. So we've got a split. So this lineage was detected again in Australia in stints in 2004, but also in 2006, it was detected in stints and gulls in Japan. And what this tells us is that probably through shorebirds, or more than likely through shorebirds, they moved this virus from Australia to Japan. Now, just because we detected it in stints in these locations doesn't mean it was necessarily moved by stints, but because it's in shorebirds, we can say it's been moved by shorebirds, and that's sort of based on some data from the US where they've been sampling a long time. Now, most of the movements of viruses along our flyway are from Asia to Australia. This is the only time we've ever seen movement from Australia to Asia. But there is also, on rare occasion, evidence of movement from North America to Australia, which is quite interesting in sort of our understanding of avian influenza dynamics in birds. Now, this is perhaps not surprising, given we know that some shorebirds go to Alaska, and then from Alaska we fly to somewhere like Australia. So they're definitely moving these viruses around. Now, these are low pathogenic viruses. They don't necessarily cause disease in birds, but the other sort of half of this story is about these high pathogenic avian influenza viruses. Okay, so when you read the news about bird flu, that's usually what we're talking about. We're talking about these viruses that tend to kill poultry. These viruses may also be zoonotic, and so they've got pandemic potential, and so we really keep a close eye on these viruses that are classified as highly pathogenic. Now, of all the HA and NA subtypes, this is restricted to 2. So high pathogenic viruses so far can only be H5 or H7. They cause outbreaks in poultry, and in some parts of the world, they're endemic in poultry. So you can sample poultry and you'll find them. And prior to 2014, these viruses always caused mortality in wild birds, such that if these viruses got out of an endemic poultry situation and got into wild birds, it tended to cause mass mortality, such that these viruses are actually very uncommon in wild birds. This changed a little bit in 2014 and again in 2016 when there was sort of a small mutation in these viruses that meant they were still pathogenic in poultry, but they were no longer killing all kinds of wild birds, and that led to the massive spread of these viruses, so which started in Asia, and then they detected them in North America, through Europe, and even in parts of Africa. So I just want to bring you up with a map here. These are all the cases of highly pathogenic influenza that are in our flyway. The little circles are either locations where there are currently outbreaks, or where outbreaks were before. I think the triangles are wild birds. And we can see sort of markings all over this map. I would just like to point out that Indonesia and PNG do not contribute data to the OIE, so just because there's no dots on the map there doesn't mean there's no action happening there. I think we can just sort of assume that there's quite a lot of highly pathogenic avian influenza in those countries as well. Now, the concern obviously is when we look at our shorebird migrations, is they're stopping in locations where these viruses are largely endemic. Now, obviously, chicken barns are not found on mudflats per se, but there is always a risk of transmission from poultry into wild birds, perhaps not directly to shorebirds, but through other bird-mediating species. And in 2016, there was actually a record of highly pathogenic H5 in a redneck stint. And so this made us think about whether or not we can detect or understand whether shorebirds that are using Australia as a wintering ground are being exposed to these viruses along their migrations. And so to do this, we're harnessing serology. So again, these birds may have been infected at some point and they generate antibodies, and then we can test for these antibodies at a later date. So all the birds in this study were sampled in Australia, but we don't have these highly pathogenic H5s in Australia. So whether we had any positives, that meant these viruses or these birds were infected elsewhere. So the figure is slightly complex. I'm just going to walk you through a small part of it, and then I'll show you the rest. So from redneck stints, we took blood samples, spun it down to serum, and then we tested the serum, which contains antibodies, against a panel of viruses, because these viruses are quite diverse. So we included 7 different strains of highly pathogenic avian influenza, in addition to 1 strain of low pathogenic avian influenza. This helps us understand whether we're detecting any H5 or specific H5s. And then each blood sample is represented as a column. So if it's white, it means we didn't test with that specific antigen, so that specific virus. If it's gray, it means we tested it and it's negative. And if it's blue, it means we tested it and it's positive. So I'm just going to show you the whole figure now. It's a bit complicated, but hopefully it's all right now. So we tested birds starting in 2011 all the way through to sort of March, April 2018. So again, every column is an individual. And we see for the most part, the birds are negative for these viruses, but there are sort of some occasions where blue does show up here, and those are positive. And we tend to see that these blue sort of bars start showing up here around 2014, which coincides well with when we had this mutation event and this new kind of highly pathogenic H5 showed up, which didn't necessarily kill all wild birds, but was still a problem with poultry. So here from 2014 through 2015. And again, we saw quite a bit of blue here starting 2016 through the end, and that coincides with sort of the second mutation event. So what we can say here is that shorebirds, like red-necked stints, that are using Australia as a non-breeding area are likely to, or are being exposed to highly pathogenic avian influenza viruses when they're not in Australia. So either on migration or their breeding grounds. And given that these viruses do kill birds, we're probably just picking up the birds that survive. We don't know the impact of this virus on birds. We don't know whether all shorebirds are surviving this, or whether it's only a small proportion that are surviving this. And I'd argue that this suggests that we really need to be doing much more work in this space here to understand the impact of a virus that causes disease like this on shorebirds as part of conservation plans, especially for shorebird species that we're seeing dramatic declines for. Now, the last sort of 3 viruses I've talked about, they all stem from viruses that we knew about in poultry. So even coronaviruses, we knew about these viruses in poultry, and it occurred to somebody to start looking for the same poultry viruses in wild birds. Again, influenza, these viruses have been known in poultry for a long time, and then we started looking in wild birds. Does it really make sense for us to continue on this path? Should we keep just sort of changing the way we target things? So we have a known poultry virus. Should we then just start looking for it in wild birds? Does that make sense? I mean, why are we looking for chicken viruses in shorebirds? That does not make a lot of sense to me. And so what we started a few years ago was starting to look at virus communities through really new and interesting methods. And a cool byproduct of this is that we started to describe lots and lots of novel birds. And so one of the key species that key taxa that we've been using in these studies are shorebirds in addition to waterfowl. And so I'm just going to walk you through some of the studies and some of the viruses that we have found. So in our first study, 2018, we looked at ruddy turnstones and avocets. I'm just going to show you the avocet data. So how this figure works is that this little avocet here, this little picture, it corresponds to something called a library. And so that just means that we pool together, or we mix together 10 samples from avocets together. And so this little avocet picture here corresponds to 10 samples from temperate Australia. This little avocet picture here corresponds to 10 samples from the interior of Australia. And the little circles correspond to the viruses that we found. So these are full genome viruses that we can pull out from the data, not little bits and pieces, but actually full genomes. And here in this case, where you see that a virus is connecting to avocets, it means it was in both of those libraries, so in those sort of merged samples. So the first thing to take away from this is, of the viruses we described here, the vast majority are novel. They've never been described before. The only thing in this study that had been described before was this duck coronavirus 2714, which I talked about right in the beginning of the talk. Now, most of the viruses here, they belong to things like Khaleesi viruses, coronaviruses, astroviruses, and rotaviruses. And prior to this study, in birds, all of these viruses were viruses that cause things like gastroenteritis in chickens. Okay, there are viruses of the gastrointestinal tract that cause gastroenteritis. But we've obviously now found versions or species of these viruses in wild birds that are healthy. They're not really showing any obvious signs of disease. Also, I just want to point out that the avocets from the interior from this study continue to blow my mind, because of the large diversity. Yes, it was 10 individuals, but they were 10 individuals caught at the same time from the same place. And there's like 9 different viruses there. And if we look at all the data, the amount of virus that they have was actually quite enormous. Sort of in proportion to all the other things, it was really high. To date, it remains the highest. And so it sort of suggests to me that avocets can carry quite a, or birds in general, can carry quite a high virus diversity somehow without really seeing any obvious diseases, quite a large viral burden. The next study we did, we were quite interested to understand whether birds that were co-captured were sharing the same virus communities. So in this case here, we launched a cannon net in Werribee, I think it was 2012, and we caught redneck stints, curly sandpipers, sharp-tailed sandpipers, and red-capped plovers together. And so in this case, we actually didn't see that they necessarily had the same viruses, and nothing as impressive as the avocets before. But from redneck stints, we found 4 different virus species. From the curly sandpipers, we actually didn't get any full genome virus species. We saw evidence there that there might have been some, but we didn't really get any full genomes. From the sharp-tailed sandpiper, we got a coronavirus, as well as this Delta coronavirus that I talked about in the beginning. This is what we now call wild bird Delta coronavirus, but at the time it was novel, and a megrivirus. And so at the time that this paper came out, of all of these viruses, the only one that had been described before was this one, which is actually a poultry relevant virus. So an enormous diversity again of viruses that have never been described before. Now, I just want to sort of take a minute to talk a little bit more about this study because it's quite interesting. So not only did we co-capture shorebirds sort of together, we also co-captured ducks together, but the shorebirds and ducks at sort of different times. And the idea was to understand whether the virus communities that are in shorebirds are different from the virus communities that are in ducks. And so, like, do you have a shorebird, a set of viruses that you consistently find in shorebirds, or a set of virus genera that you find that are important for shorebirds and a set that are important for ducks? Or are they mixing a little bit more? Which is quite an interesting and reasonable question, I think. And so this is one of the figures from the paper. I'll just walk you through it. So these are all the species of birds that we looked at. So again, these 4 were sampled together, same place, same day. Those 5 were sampled together, same place, same day, but different places. And they're arranged here on the left by their host genetics. So this is how these birds are related to each other and how these birds are related to each other. And then on this side, we've sort of made a tree of our communities. So essentially all it means is that sort of like great teals and Australian shelducks, because they're connected like this, that means that their communities are quite similar to each other, whereas sort of they're more distantly related to communities from here. Now, if we had expected to see that the viruses from shorebirds and the viruses from ducks were sort of discrete units, we would have seen that sort of no crisscrossing here. We would have just sort of seen green on the bottom and blue on the top. But what we actually see in this figure is the crisscrossing, and that sort of suggests that the virus genera in this case that are found in shorebirds are also found in ducks. So we're seeing probably more generalist viruses than we're seeing specialist viruses, which is perhaps not surprising, given I talked about avian influenza before, which is found both in ducks and shorebirds, and that coronavirus called duck coronavirus 2714, which is also found both in shorebirds and ducks. So this helps us understand that if we want to understand or have a look at viruses in shorebirds and the impacts of viruses in shorebirds, we also perhaps need to look at some other species like ducks, which may be a reservoir for viruses that occasionally spill into shorebirds or things like that. The study that we have was just submitted this year. We used ready turnstones as a model species to try to understand whether there are differences in virus communities based on bird age. So this figure is a little bit more complicated than the others. So here in the columns is the age. So these are 1+ birds. So again, 1 bird is 10 samples combined. So it's a library and a library. We have 2 0 bird here, and we've got 3+ birds here. These birds were sort of co-sampled together in November. These birds were co-sampled in the subsequent March. And so again, the vast majority of birds or of viruses we found here were novel. Now this is surprising because we sampled the same location as we've sampled before. We only sampled one host species here. So it's amazing that in just ready turnstones at 2 time points, we found 14 different virus species, 12 of which have never been described before. And again, that coronavirus 2714 keeps showing up. So this is obviously an important virus for shorebirds. And we found another one called the Wille virus too. And the other really interesting result here is that we found that juvenile shorebirds, so first-year shorebirds, have a larger virus diversity than adult birds. Okay, so you can see sort of around this shorebird, there's sort of more little virus circles here than there are around adult birds. And if we look at in the data at sort of like how much of the data is devoted to viruses as compared to things like the microbiome of the host, again, we see that the viral load in young birds is also much, much higher than in adults. So young birds are being exposed to quite a lot of viruses. Now, this is perhaps not that surprising if we think about humans. Kids in daycare are like little virus factories. So that's not surprising when extended to things like wild birds. But I continue to be amazed that every time I do another study in a location where we've done a few studies in birds that we've sampled quite heavily, that we just keep finding this incredible diversity of new viruses. Now, just because you find a virus, what does it mean? And so this idea of viruses causing disease is an old idea, and I addressed this right in the beginning. Just because you find a virus doesn't necessarily mean it causes disease. And so I just want to show you a quick example here. So this is part of a tree from the Khaleesi viridae. The Australians in the audience would know these viruses. This is what we're using to control rabbits. The other good example is if you've ever been on a cruise ship and gotten noro, that's a Khaleesi virus. So in 2008, if we looked at all the Khaleesi viruses known in birds, they clustered together, but they're all from poultry, and they're all from studies where they found gastroenteritis in poultry and wanted to know what it was. So it's Khaleesi viruses. In 2020, so just including sort of 3 of our publications, we've now increased the number of Khaleesi viruses quite dramatically. And all these Khaleesi viruses that we found were found in seemingly healthy wild birds. Okay, so now what I would argue is that Khaleesi viruses are not necessarily associated with disease. It's rather actually that Khaleesi viruses in poultry are the exception here. But there are viruses that we know that do cause disease, okay? And so this is that sort of story of highly pathogenic avian influenza. And while we know there may be evidence of it in shorebirds, we have no idea what the burden is. And so there is obviously space to do work here to understand the burden of viruses on shorebirds, whether it's those that don't cause disease or those that do. And just to end this, I just want to end this on one sort of quick note, and that's even though we have viruses that don't necessarily cause disease in birds, there's quite a stable relationship there between shorebirds and their viruses. Now, we are in a changing world, and this changing world may modulate this relationship between shorebirds and their viruses. So for example, we have relationships with the bacteria and viruses that are around us all the time. But if we as humans become immunodeficient or have an immune problem, we may actually fall susceptible to viruses and bacteria that live in our skin and that are part of our healthy microbiomes. And one of the concerns for shorebirds is that this might be the case. So this is a map from a paper a few years ago. In this case, white is the continents, and the color there corresponds to levels of pollution. And so I'll just sort of overlay this again. You guys all know the migration routes of shorebirds, but again, we find that places where shorebirds stop are places where there's really high levels of pollution in the environment. And we know from studies in people and other organisms that pollution may modulate the immune system. And if you have the modulated immune system, this may affect the relationship that you have with your viruses, and this may sort of increase disease burden. And so this is really exciting research that's being led by Marcel Claassen at Deakin, Fjorla Jespers in Norway, and a number of students. And that's just sort of something for us to think about as we move forward. So in sum, with the exception of low pathogenic avian influenza virus, we know very little of the ecology of viruses in shorebirds. We have a very poor understanding of the diversity of viruses that are found in shorebirds. This ties into the next point, where it does not necessarily make sense for us to continue focusing only on poultry-relevant viruses in shorebirds. And of the viruses we find, the impact on shorebirds is extremely unclear, but the vast majority probably do not cause clinical disease. And things like pollution may have a negative impact on the shorebird virus relationship. And I'd also like to argue that I think it's time for us to really start considering things like viruses in our sort of conservation plans of species that we're seeing dramatic declines for. And with that, I'll take questions. Thank you.